All right, let's finish up our microarray experiment here. So the first thing we're going to do is we need to create our groups that we loaded in with the file before, our targets, so it can be read by limma, which is our differential analysis package. So we're going to call groups, and that is our targets, and it's the second column. So remember, if we look at our targets, we want this column to be what distinguishes if they are ground or flight. So we'll run that line. Okay, so we have our groups here. We have ground and flight, perfect. And now we're going to modify groups. So if we want to modify a variable, like we don't need this anymore. We want groups, but we just want to modify something about it. So we're going to call this a factor. So as you can see here, is used to encode a vector as a factor, kind of like a category. And we are going to then say that those values are factors, and there's going to be two levels. So there's two distinct categories within them, and they are going to be ground and flight. So let's run this line. Okay, so now you see we've changed it to a factor with two levels, ground and flight. And then it gives us this matrix here that tells us which rows are part of ground and which rows are part of flight. So it's kind of like a dummy coding, as they call it. So now we're going to create the design of our experiment. And we're going to create a model matrix. So it creates a design by expanding factors to a set of dummy variables, which that's what these are, right? Dummy coding. And for our annotation, we're going to say by 0 and groups. So we're going to modify it or look at our the difference in these groups. This is what this is saying. And then let's change the column names of our design. And our column names, because right now our column names are just x1 and x2. So we're going to label them the same thing. We're going to label them ground, and, I'm typing and like I'm annotating. Flight. Oh, we should probably annotate, speaking of. Here we do our differential gene expression analysis. Okay, so I'm going to run all these. I'm just going to run them over real quick. Okay, perfect. All right, so now let's actually, I'll make a new chunk for this. And we'll call this fitting the linear model. So our fit is going to be LM fit, so linear model fit, of our expression set that we had earlier, right up here. And we're going to do this by our design that we set up to look between groups. And then we're going to make a contrast matrix with make contrasts of ground versus flight and ground minus flight. So contrasts are very important when you are comparing more than two experiments. It's a good practice to do contrast. So say you have a 2x2 experiment and you have, say we did flight and ground, and then we did ground in the light and ground in the dark, right? So we have light grown ground, dark grown ground, light grown space, dark grown space. These contrast matrix help to kind of normalize these regression models and get rid of the variance between experiments. So it's good practice to do that. So now fit contrasts fit, and then our cont matrix. All right, so now we're just doing adding our contrasts to our model, and we're going to do a Bayesian adjustment, which helps to so given the linear model, it computes moderated t statistics and f statistics. So without, it's a rabbit hole to get into Bayesian theory, but Bayesian theory kind of is all based on densities and not necessarily discrete like p-values and things like that. But this is a way to adjust our computation of differential expression. If you'd like to know more about Bayesian statistics, I'm sure YouTube would have plenty of videos on that. So now we're going to look at our results. So we've adjusted our linear model, and we're going to do decide tests of fit. So this is, whoops, where am I? There we go. Decide tests of fit. So decides tests is going to look and do our differential gene expression analysis. See, does it? So let's see what it says here. Identify which genes are significantly differently expressed for each contrast from a fit object containing p-values to test statistics. And then we're going to adjust the p-value in a second with the second part. So this is going to tell us what is significantly different between space and our ground control experiment. All right, and now let's write our results. And we're going to call this brick16.txt. And this is where we're going to adjust add our adjusted p-value. So we're going to write adjust, and we're going to do, see if it tells us what our options are for adjust. It doesn't. And we're going to use BH, and give me 1 second here. I want to quick check something. So I wanted to get this right because I think I've messed up their names before. So this adjustment for p-values. So the reason we do an adjustment, let me take a step back, is because when you do so many tests, then there is a possibility of getting a false positive. So let me pull up this as a quick example. So this cartoon kind of does a good job of showing it, right? And so this person says jelly beans cause acne, and the scientist goes to investigate with a bad dad joke about Minecraft. And so he says, we found no link between jelly beans and acne. And so P is greater than 0.05. So a way to think about this is that a p-value of 0.05 is, in essence, the amount of false positives you're willing to accept. So great. So in this, you're a statistician would not like the way I'm going to explain it, but I don't want to get too much in the weeds. So you're allowing for 5% false positives. And so the scientist says that settles it. And then the lady says, I hear it's only a certain color of jelly beans that causes it. And so then they found no link between purple, brown, pink, blue, teal, salmon, red, turquoise, magenta, yellow, gray, tan, cyan. Green, they did find it. Mauve, beige, lilac, black, peach, orange. But because they didn't do multiple test correcting, because they did so many comparisons, eventually, say you did 100 tests of jelly beans, if your p-value is 0.05, then 5 of them theoretically would come back positive just by chance. And so then the news article says green jelly beans are linked to acne. So when you do a ton of, the moral of the story is when you do a ton of these tests where we're doing 22,800 or whatever number of genes that we're testing between, you have to have multiple test correction, because theoretically, if you didn't, then 5% of those genes would come back as differentially expressed just on chance. So what we use is this BH adjustment, which stands for Benjamini and Hochberg, who wrote a paper, I can't find the date, and found a way to increase the stringency to account for how many tests you're doing. So always great, like if you're doing, a large amount of comparisons, always use multiple test correction, because that is important so you don't get green jelly beans. All right, I'm gonna run these. Uh-oh. That should be a period, I believe. Let's try this again. Bays. What am I misspelling? Because it's always got to be a misspell. B-E-Y? Is that what it is? No, B-A-Y-E-S. Let's try it again. There we go. All right, so we're writing our fit. Okay. And so, let's look at what this looks like. What I call brick16.text. Beautiful. All right. So it's probably makes more sense to open this in Excel, so your columns kind of line up better. But as you can see, we have our coefficient, which is the change in expression. We have, all right, hold on. Or no, our coefficient would be this column. So we have positive and negative changes in expression. But what's important is this last column here, this column right here. This is our FP value, sorry, that's our F value. Where's our, all right, let me open this in Excel. Enough trying to get it lined up so we can see what's going on. All right. Let's try this again. I'm gonna browse. NASA. Yeah, change this because it's only looking for Excel files. So we have brick16 text. And it's going to ask us to convert that so we can see what's going on. There we go. All right. So here's our adjusted p-value. And so we would be interested in p-values that are less than 0.05, right? So we are going to sort, and we're going to sort by column p-value adjusted, smallest to largest. And so as you can see, these are all significant. These genes are all significantly different. And so if we were to go down, we would have 0.05 would be our cutoff. So we would have 1,351, because we have that first row as our header, genes that are differentially expressed between Earth and space. Just to take a peek. Okay, so let's keep going here. So now let's select those genes, right? So we want to select only those genes that have a p-value of less than 0.05. Oh wait, we fit. Hold on. Okay, so we're not doing, we don't have p-adjust in here, so we're just going to look at the p-values unadjusted, which, but we're just doing this for graphing purposes. Okay. All right, so now let's select our, out of that ESET data frame, our ESET, and we're going to say selected. So this is out of our expression set. We're taking only the our list of selected. So if we run this, we have, it goes through and it's a logical, and it says, it reads every line and it says true or false if it's less than 0.05. And now we use that list and we take all those trues out of our ESET, this big data frame. So let's do that. Okay, so ESET select. So we have our expression set for ESET select now. Let's continue. All right, so let's pull out the coefficients. These are the expression, average expression for flight and ground. All right, let's run. Okay, so these are all the fold change for our flight versus our ground for each gene. So with ground being control, let's look at this. So this gene is negative. So this gene is turned down in space. This gene's turned down in space, down, down. This gene's turned up in space, down, etc. Okay. Now let's do so we're gonna add, let's look at fit again real quick. Debating if I want to change this. All right, so we're going to use Oops. P.val. Oops. I have insert on. Right, it's called p-value. Maybe we'll check one more time. P.value. All right, so we're going to add our p-values back to our into this variable. So now we just have a list of all of our p-values for our experiment. And for final, we're going to do cbind all coefficients p-val. Actually, let's try something here. I want to look at keep looking at fit. All right, let's write this real quick. All right, bear with me here. I'm having some ideas. I'm gonna try doing this real quick. Okay, well, I wanted to grab the adjusted p-values because these are just the normal p-values. We have them here in our written file, but this is just the normal ones. So you have them, they're just not in a place. What we could do is we could, because this right fit doesn't save anything, we could reload this and then just take that column and add it. You know what? Let's do it. Let's be thorough, everybody. All right. This is probably not the best way to do it, but I don't want to look it up while you guys are stuck here watching me make this video an hour and a half. Okay, so we're going to read this brick 16.txt quick, and then sep equals, what was that? Tab, I think. All right, let's read that real quick and see what happens. Adjusted. Okay, so now we want to grab, instead of grabbing, we want this p-value adjusted. So instead of grab for the cbind, I'm going to move this down here. We're going to add adjusted, and we're only going to add the p-value adjusted. So let's try this. Actually, let's add both so we can compare them real quick. Oops, didn't even run all that. All right, let's look at this real quick. All right, so we got there we go. Our row names, which are the same, we probably didn't need that, but our gene, KEGG, gene ontology, the gene symbol, all this great data, our expression for each individual microarray, and we have our ground versus flight coefficient. I don't know why it glitches like that. And then we have our p-value adjusted. So this is our expression change. This must be our original p-value. Is that from, here's p-value. Look at this real quick, 0.0069. You're getting a real look into what creating a pipeline is. Okay, so this is our p-value, unadjusted, and then this is our p-value adjusted, because that this 0.0069 matched our p-value, and this is our full change between flight and space. Okay, quick detour on my planned lecture, but hey, we got it. All right, now the last thing we're going to do. So that's all of the data, right? So we have our changes in expression between ground and flight. We have our what ones are significantly different between flight, our original p-value, we adjusted our p-value, etc. We've annotated it all so you know what genes are actually what. Beautiful. The last thing we're going to do is we just want to kind of take a look back at our experiment and see to make sure the groupings of our experiment make sense, in that how similar was the expression changes in all three of those flight experiments or flight chips versus the 3 ground controls. And I added this at the end because it can take a little bit of time. We're going to cluster it by DE pattern. And then we are going to say the column equals null. We're going to say the linkage row equals equals complete. Linkage column equals complete. I just want to I'm not getting too show dendrogram equals both. Not too much into the variables because I don't want this video to be super long, but I just want to show you that this is something we can do. 0.05. Okay, so this is going to create 1 decimal place. All right, so this is going to create a oh, no comma here. Okay, let's try this one more time. Null. And how about with an uppercase U? That's what happens when I rush. Okay, so like I said, this is going to take a little bit of time, but I already have it done. So what this cool map shows is it shows the grouping of the experiments. So if you look down here, we have our ground 2, ground 3, and ground 1, and then flight 3, flight 1, and flight 2. And what we want to see is similar changes in expression of these genes based on the experiment. And so we've created a dendrogram, so kind of like an evolutionary history, right? Where we see that these 3 flight experiments, they group together. And we see that these 3 ground experiments, they group together, right? So this is really good to be able to see that, yes, the expressions seen in the 3 ground experiments were more similar to each other, and the 3 flight experiments were more similar to each other than their other groups. And that's what we really want to see, because if ground 1 was more similar to flight 2, then we might have a lot of noise or error in our experiment. So there is one more thing I can show real quick. So if you do want to look at just the top, say 10 genes and their adjusted p-values, you can call this top table of your fit, and it will list the genes, the log fold change, average expression, p-value, adjusted p-value, etc. But I'm not going to run that because this is probably going to take like 5 minutes to run. So that is it. We're done with microarray experiments. We will keep your data and stuff. We will eventually also do some pathway analysis on this data, I think. But I want to do RNA-seq first, and then we can do pathway on both of them to kind of look at it. But yeah, so that is it for microarrays. Hope you enjoyed it and learned something. If you want, you can dive into the data that you generated and see what genes are upregulated and downregulated, and it might be what you expect, it might be what you don't expect. But that is it, and I will see you in the next video.